Hear me okay?
Yeah. Give a warm welcome also.
Thank you, Morton, Daniela, and Alex. I'm very happy to be part of this yet another exciting meeting. So some of you may know that my lab has been taking human genetics and functional genomics approach to understand the basic mechanisms of aging in humans. Thank you, Jennifer, for shout out during your fantastic talk. Recently, I started human genetic studies of ovarian aging, but that's not what I'm going to talk about today. I made a brand new talk that I have never given before. So let's see how it goes. But we've been working on establishing heterochronic parabiosis system in vitro and identify geroprotective factors from young blood. So we heard from Tom Rando yesterday and Sol Vieda today that heterochronic parabiosis studies have convincingly demonstrated that young blood harbor rejuvenation factors that has many beneficial effects in aged body. So Tom's pioneering work on muscle and liver, and then the list goes on, and it really has a beneficial impact on the variety of tissues, including brain, you heard from Sol, kidney, heart, bone, and we cannot hardly keep up. And the opposite is also true, right? The old blood has deleterious factors that prematurely age or cause age-related dysfunction in multiple tissue types. The big question is, what are these factors that mediate this effect? And thus far, only a few factors have been identified, maybe largely because they have been tested in animal models, primarily in mice, which takes time, costs money. So we've been talking about establishing heterochronic parabiosis system in vitro so that we can screen candidate geroprotective factors from young blood or vice versa, old blood as well. So this work has been led by three very talented and dedicated people in the lab. Adam Hudgens and CJ Wang are former graduate student at Einstein, and they're now postdoc in my lab at Columbia. Chadash Tajaslan was a postdoc at Einstein. So whenever I say we, I mean these three people. And Adam and Chadash is now in the process of incorporating company based on this work. It's called Perpetual Bio. And they are here. Maybe you have questions, and they will be happy to answer. So when you establish this in vitro system, of course, big question is, what are the endpoints, cellular endpoints, that may predict or recapitulate geroprotection in vivo? We base ourselves on two major discoveries made in the lab. Number one, when you look at human primary cells, in culture they have finite replicative lifespan. So these are 4 primary lines. And they enter cellular senescence at one point, and they don't divide anymore. But geroprotectors such as rapamycin extends replicative lifespan, thereby delaying cellular senescence. Number one. Number two, last year at ARDD, I talked about 3 longevity-associated variants that I pulled out of near centenarians. And when we model them in primary cells by using CRISPR knock-in, they really extend replicative lifespan and show some resilience to cellular senescence. So based on these 2 discoveries, we concluded, okay, cellular senescence, resilience to cellular senescence, and or extension of replicative lifespan can be used as a cellular endpoint, of geroprotection. So how do we mimic a heterochronic system? So first, we tested age of serum. So as you know, the standard cell culture, we use fetal bovine serum known as FBS. And then we started to culture them from serum with increasing age. And then you can immediately see already in 1.5-old serum from this bovine has adverse effect and cause early senescence, and it gets worse when you reach 6 years, and by the 10 years, it's delayed. So lifespan, replicative lifespan is shortened. And this has been confirmed by many markers of cellular senescence, including expression of cellular senescence-associated genes and beta-gal, as well as proliferation markers such as Ki67. And this response to aged serum is really very, very abrupt. So only 24 hours after you switch serum from FBS to like 10-year-old serum, so serum from 10-year-old bovine, and it caused enormous changes in gene expression. Some 800 genes are differentially expressed, and when you look at them, they are enriched in cell cycle control and nutrient sensing, signaling, and inflammation. And you can also see them are enriched in San Mayo senescence gene panel. So next question is, how do you mimic this heterochronic condition? So as you may predict from in vivo heterochronic parabiosis, we simply mixed serum from young adult and serum from old adult. And then using that mixed serum, what we call heterochronic culture system, we apply to senescence assays. And then you can see that in heterochronic condition, this young serum relieves some symptoms of this age-related changes, right? So it increases reproductive lifespan compared to just aged serum only. Again, this was confirmed by other markers of senescence. So this is bovine sera. What about humans? Because eventually we want to translate the finding into human situation. So we used human sera, and then we found two things. Number one, they are not as supportive as bovine serum. So you can see that even young serum from like 20, 30 to 29-year-old people do not support proliferation very well. So, population doubling is less than 10 compared to bovine is like 20. But nevertheless, adding young sera to old sera can improve the endpoint. That's number one. But these results are from male serum. But female serum, the old serum does not really support growth very well. But nevertheless, when we add back young serum to aged serum, we could promote proliferation, suggesting that at least in part we can recapitulate what's happening in vivo heterochronic parabiosis condition in this culture system. So question was, who are these factors in mediating this young versus old serum effect? So as you know, the serum has many, many components. So we wanted to prioritize where to look first. So we dialyzed to remove metabolites, small amino acids, micronutrients. We also depleted exosome so that we remove microRNA and exosome cargo. We also did the charcoal strip so that we remove some protein, lipids and hormones. And then we also use temperature, high temperature, two different temperature to partially or irreversibly denature protein. And then apply this serum, different fractions of serum, to our heterochronic conditions. And then you can see that except for dialysis, in all this different manipulation, reduces reproductive lifespan. But the strongest effect was found when we denature proteins. I mean, especially when you completely irreversibly denature protein at 95, you really don't see much. It really causes premature senescence, right? And that can be also measured in other markers of senescence. Okay. So we decided to focus on protein factors based on our data. And so who are these protein factors? So we did unbiased proteomic analysis of, from young adult to old, and then identify some 650 proteins that changes in abundance with age. And to further prioritize, we had two different criteria. Number one, of course, we have translation in humans in mind. So we wanted to see this conservation in humans by looking at this large human cohort plasma proteomic data, here, and see at least the directional changes with age in abundance matches, okay? That's number one. Number two, as Near and Steve elaborated yesterday, geroprotectors, if it really has to have geroprotective effect, it must have pleiotropic effect, right, to just sustain health. So we also look at potential multiple tissue effect. And PEDF was our top candidate because it fits the bill in both criteria. So its abundance in circulation declines with age, both in bovine and human, and also many different tissues in human body based on GTEx analysis. And it has really multi-trophic, multi-protective role in many different tissues. So what is PEDF? PEDF is pigment epithelium-derived factor. The name actually came from this conditioned medium of RPE, that's retinal epithelium, retinal RPE, pigment epithelium, right? And it really has proliferative potential. I mean, the proliferation and differentiation of multiple tissue-specific stem cells, and it has beneficial impact in many organs, including the brain. It, promote muscle regeneration, osteoblast differentiation. It also have anti-inflammatory and anti-tumoric, anti-fibrotic activity. So this was our top candidate, and we tested using recombinant PEDF. And in our heterochronic system in culture. So first of all, when we take human serum from old people like 85 years, it doesn't really promote survival of the cell, but when we add PEDF, then you see the significant improvement in cell viability. And in this condition with the 85-year-old serum, additional PEDF improve replicative lifespan. And when we neutralize PEDF using neutralizing antibody, then it reduces effect of young serum. This result suggests that PEDF is sufficient and necessary for this protective effect of young human serum on these cellular endpoints that we are going after, replicative lifespan in human primary fibroblast. So next question was, what about in vivo? As we predicted or hoped, is this factor has multi-tissue restoration effect in this age-related dysfunctional body in animals? So I'm going to show you several examples. So first of all, we tested in brain, as you heard from Sol, and also Tom yesterday, that brain is one of the main target of young blood in heterochronic parabiosis setting. So what we did was we took aged mice in their 20s and systemically supplement PEDF using subcutaneous osmotic mini pump. And then we started to measure behavior in 5 weeks, altogether treated for 6 weeks. And then this hippocampal dependent memory assays, novel object recognition assays, you see that PEDF actually significantly improved this novel object preference, and it immediately induced changes in gene expression, and they are enriched in biologically relevant categories such as synaptic transmission. And you can see genes like TIM2, which is known to improve cognition in the context of heterochronic plasma transfer, is upregulated in response to PEDF treatment. In the liver, it reverses liver fibrosis, and it causes changes, mostly gene expression, mostly enriched in metabolism. And you can see that genes, transcription factor like FOXM1, which is involved in regulation of liver regeneration and recovery from injury, is upregulated. And the catalytic subunit of AMP kinase is also upregulated in response to PEDF treatment. What about kidney? In kidney, PEDF administration reduces age-related lipid accumulation, and then you see that gene expression changes induced by PEDF, which are enriched in ATP synthesis, mitochondrial function, also genes involved in macrophage biology. And then you can see these genes like TREM2, which is involved in macrophage-mediated scavenging of lipids, are upregulated. I mean, these are also involved in the pathway, suggesting that maybe it contributes to rejuvenation of the kidney by protecting them against age-related lipid accumulation. And we also look at ovary, because ovary is one of the very first organ to age in the body. So in this case, we use reproductively old age. It's 11 months instead of 20. And then we treat them with PEDF for 4 weeks. And then we see that PEDF treatment without affecting primordial follicle proportion. So it's not going to affect the activation, but it's going to promote follicle growth as measured by Ki67, the percentage of growing follicles higher than control, and also percentage of dying follicles is small. So rather than involved in follicle activation, it may be involved in follicle maturation and growth, thereby may contribute to this protection against age-related decline in fertility. So what about disease model? As I said, brain is one of the best-known target of heterochronic parabiosis of young blood. And so we use, there's a study showing that young plasma improve cognition in Alzheimer's disease patients. So there are several clinical trials are ongoing. But of course, the factors are not known. So we use this 5xFAD AD model, which carries this early onset Alzheimer's disease, and tested their cognitive function using Y-maze, and we see improvement. Already at 12 months, where this amyloid pathology is really very high, the burden, and you start losing neurons. Finally, and then they PEDF improve blood-brain barrier integrity in this Alzheimer's model. Finally, when we look at high-fat diet-induced obesity, PEDF treatment also protect against weight gain. I mean, this is old mice, 18 months, that have been under high-fat diet for 3 months. So with that, I would like to summarize my talk. So we established this in vitro heterochronic parabiosis system to efficiently screen candidate geroprotective factors from young blood. And then we identify candidates through unbiased proteomic analysis of young blood, and further prioritize based on overlap with the humans and potential function of the protein in multiple tissues. And PEDF was our top candidate. And then we test back in our in vitro system to confirm it, at least it mediates the effect of young blood. And then in vivo, we tested that the supplementation of PEDF has beneficial effect in many tissues, including brain, liver, kidney, and the ovary, as well as Alzheimer's disease setting and the high-fat diet-induced obesity. So we believe that this paradigm can be used to screen and identify potential geroprotective target from young blood or anti-geroprotective target from old blood. With that, I'd like to thank Adam and CJ, together with others, carry out the work, and Chadash, who was deeply involved in this work. And I'd like to thank Tom for collaboration and guidance and mentorship and funding from NIA. Thank you for your attention.
Thank you so much, Yousin. Do we have any questions? We have time for maybe one short question, maybe from Anton. Wait, you have to speak in the microphone, otherwise they can't hear you at home.
So what I didn't understand, like in the very beginning, you had like comparison between how cells were doing after like FBS, 6 and 10 years, and I think that 6 years were doing worse than 10 years.
No.
I don't know, I saw this chart. Okay, maybe I misseen it. And actually, and also for PDF, how much does it extend lifespan on model organism? Do we have such data?
Oh, yeah, thank you for the question. So PDF itself, the ortholog is not well characterized in the animal models, but it's a receptor. I mean, it has many receptors. One of the major receptors, ATGL, in C. elegans, when you overexpress it, it extends lifespan by 30%. And in flies, when you overexpress, it improves health span in different measures. So it clearly has beneficial effect in animal models. But nothing is known in mouse models.
All right, thank you so much, Yousin. I think we have to move on in the
Thank you.
interest of time. Let's give her a round of applause.